Which woods are used for telephone poles?
The principal woods used for telephone poles are southern pine, Douglas fir, western red cedar, and lodgepole pine. Ponderosa pine, red pine, jack pine, northern white cedar, and western larch are also used.
How much wood is needed to make 1 ton of paper?
In the United States, wood pulp is usually used in paper manufacturing. Pulp is usually measured by cord or weight. Although the fiber used in making paper is derived overwhelmingly from wood, many other ingredients are needed as well. 1 ton of paper typically requires 2 cords of wood, 55,000 gal, 208,000 L, of water. 102 pounds 46 kilograms of sulfur, 350 pounds 159 kilograms of lime, 289 pounds 131 kilograms of clay, 1.2 tons of coal. 112 kilowatt-hours of power, 20 pounds, 9 g, of dye and pigment, and 108 pounds 49 kilograms of starch. Other ingredients may also be necessary.
What is lyophilization?
Lyophilization is a freeze-drying technique for preservation and storage of bacteria and other microorganisms. Bacteria can be stored for extended periods of time, 3 to 5 years, as frozen cell suspensions or as freeze-dried, lyophilized, cultures. This technique is achieved by placing bacteria in a nutrient broth containing 15 to 25% glycerol and freezing at temperatures of -94 degrees Fahrenheit -70 degrees Celsius or lower. The glycerol reduces ice crystal formation that would cause subsequent cell damage and disrupt biological structures.
What is a vector?
A vector is an agent used to carry genes into another organism. Specific examples of natural vectors include plasmids or viruses. In human gene therapy, vector viruses must be able to withstand the challenge of the patient's immune system. Once the vector manages to invade the immune system, it must be able to penetrate the cell membrane and, finally, must be able to combine its genome into that of the host cell. Vectors are also crucial to plant and animal genetic engineering.
What is the difference between rough and smooth endoplasmic reticulum?
Rough endoplasmic reticulum are regions rich in ribosomes that manufacture proteins. These regions appear to have a pebbly surface that is somewhat similar to sandpaper. Regions with no ribosomes are referred to as smooth endoplasmic reticulum. The smooth endoplasmic reticulum is involved in the synthesis of lipids and steroids, carbohydrate metabolism, and the inactivation and detoxification of drugs and other components that may be toxic or harmful to the cell.
Who was Dian Fossey?
Dian Fossey, 1932 to 1985, was an occupational therapist who, inspired by the writings of the naturalist George Schaller, 1933, decided to study the endangered mountain gorilla of Africa. She was trained in field work by Jane Goodall and went on to watch and record the behavior of mountain gorillas in Zaire and Rwanda. She eventually 326 obtained a PhD in zoology from Cambridge University and in 1983 published a book on her studies. Gorillas in the Mist. In 1985 she was found murdered in her cabin in Rwanda. Her death is still unsolved.
How do chromoplasts differ from chloroplasts?
Chromoplasts are pigment-containing plastids found in plants. They are responsible for the characteristic red, orange, or yellow coloration visible in some flowers, fruits, and plants.
Are some forms of cancer communicable?
Yes and no. Cancer cells arise from within the body. However, certain types of cancer may actually be caused by viruses, which may be transferred from one organism to another. Currently, it is estimated that viruses may play a role in as much as 15% of human cancers worldwide.
How does the iodine test detect starch?
Starch is a coiled polymer of glucose. Iodine reacts with the coiled molecules and turns bluish-black when added to a solution. A solution that remains a yellowish-brown color is a negative test for starch, whereas one that turns bluish-black is a positive test for starch.
Is coffee native to Colombia and Brazil?
Although premium coffee is today grown in the mountains of Central and South America, the coffee tree, Coffea arabica, is native to Ethiopia. Coffee was widely used in the Arab world before its introduction to European society in the 17th century. North and South American coffee plantations were started in the 18th century.
What is habituation?
Habituation is the decreased response to a stimulus that is repeated without reinforcement. Habituation can be very important to an animal in its natural surroundings. As an example, young ducklings run for cover when a shadow, a possible predator, passes overhead. Gradually, however, the ducklings learn which types of shadows are dangerous and which are harmless.
How much do plants contribute to the human diet?
In the United States and Western Europe, approximately 65% of a human being's total caloric intake and 35% of consumed protein are obtained from plants or plant products. Soybeans are an example of a plant with high protein content. In developing nations, almost 90% of calories and more than 80% of protein in a person's diet are from plants.
In what ways are earthworms beneficial?
Earthworms help maintain fertile soil. An earthworm literally eats its way through soil and decaying vegetation. As it moves about, the soil is turned, aerated, and enriched by nitrogenous wastes. Charles Darwin, 1809 to 1882, calculated that a single earthworm could eat its own weight in soil every day. Much of what is eaten is then excreted on the Earth's surface in the form of casts. The worms then rebury these casts with their burrowing process. In addition, Darwin claimed that 2.5 acres, 1 hectare, of soil might contain 155,000 earthworms, which in 1 year would bring 18 tons of soil to the surface, and in 20 years might build a new layer 3 in 11 centimeters thick.
What are the steps to prepare a specimen for examination?
The 3 basic steps to prepare a specimen are fixation, preservation, staining, and mounting. Preservation prevents destruction or decay of the specimen as well as inhibiting microbiological growth. Different stains and dyes attach to different parts of a cell, such as the nucleus. Specimens are mounted in a medium that is also a preservative and often covered with a cover slip.
What is the relationship between bacteria and temperature?
All microorganisms have temperature ranges that determine growth. Overall, microorganisms are unique in their ability to exist and grow at temperatures ranging from 14 degrees Fahrenheit to 230 degrees Fahrenheit, minus 10 degrees Celsius to 110 degrees Celsius. Temperature restrictions are due to limitations in cell metabolism. A microorganism's maximum temperature is the highest temperature at which growth can occur. Minimum temperature is the lowest temperature at which growth can occur. A microorganism's optimum temperature is the temperature at which the growth rate is the fastest. The maximum, minimum, and optimum temperatures define the range of growth for each microorganism and are collectively referred to as the cardinal temperatures. Bacteria are divided into 4 groups on the basis of their cardinal temperatures for growth: psychrophiles, mesophils, thermophils, and extreme thermophils. The following chart lists the temperature ranges at which these groups can grow.
How can bacteria survive inside our gut?
Bacteria known as intestinal flora survive in both the small and large intestine because of the neutral pH of the intestinal environment. Because these bacteria have low requirements for oxygen and sunlight exposure, they are well suited to life in this environment. They use our digested food as a source of nutrition and even provide a benefit to us. By synthesizing several of the vitamins, biotin, as well as vitamins K and B5, we need. If the flora are thriving, it is more difficult for disease-causing microbes to establish themselves in the intestines and attack our bodies.
Are Rickettsia and Chlamydia bacteria or viruses?
For many years, Rickettsia and Chlamydia were thought to be viruses because they are very small and are intracellular parasites. They are now known to be bacteria because they possess both DNA and RNA, have cell walls similar to those found in gram-negative bacteria, divide by binary fission, and are susceptible to antibiotics that produce an effect in most bacteria.
How many species of fungi have been identified?
Scientists have identified between 70,000 and 80,000 species of fungi, almost 2,000 new species are discovered each year. Some mycologists estimate that there are about 1.5 million species worldwide, placing the fungi second only to insects in the number of different species.
How does the cuticle protect plants?
The cuticle contains a waxy substance, called cutin, that covers the parts of the plant exposed to the air, the stem and leaves. It is relatively impermeable and provides a barrier to water loss, thus protecting the plant from desiccation.
What role do bonds have in bioorganic molecules?
Bonds are important to the structure of bioorganic molecules. Because chemical reactions actually involve electron activity at the subatomic level. Shape determines function. For example, morphine has a shape similar to an endorphin, a natural molecule in the brain. Endorphins are pain suppressant molecules, thus. Morphine essentially mimics the function of endorphins and can be used as a potent pain reliever.
What is meant by a codominant allele?
Codominance is an example of non-Mendelian genetics. When alleles are codominant, all versions are expressed in the phenotype. An example of this can be found in chinchilla rabbits, where there are 4 genes that affect coat color.
What are different ways of defining the term species?
A species may be defined in a number of ways. The biological species concept defines any 2 individuals that can breed and produce fertile offspring as belonging to the same species. This would mean that lions and tigers would be considered the same species. As they can produce hybrid offspring, there is at least one case of a tigon. The offspring of a male tiger and a female lion, producing offspring after an unplanned mating with the tiger. Other concepts include the phylogenetic species concept, which bases species determination on the shared evolutionary history of the populations in question.
What is sexual selection?
When individuals of the same sex differ in their mating success and that difference is correlated to the presence or absence of a particular trait, that trait is said to be the result of sexual selection. Traits that provide no benefit to survival but that increase the likelihood of the male acquiring a mate are examples of sexual selection.
What are examples of courting behavior?
Male stickleback fish swim in a stereotypical manner as they court potential mates. Male bowerbirds build elaborate towers of vegetation to entice females. Female moths release pheromones that attract males from up to a mile away. Male African elephants use low-frequency sounds to find females who are sexually receptive. And, of course, male birds and frogs use vocalizations to attract mates.
Is it possible to observe evolutionary change in other species?
The change in allele frequencies within populations, which is driven by natural selection, is occurring around us constantly. However, whether we can actually observe those changes is a different matter. For example, elephants may be adapting to changing conditions in their environment. But since they tend to be extremely long-lived, it is unlikely that we will be able to follow enough generations to observe a trend. On the other hand, it is possible to notice such changes in populations of individuals with very short lifespans, particularly when they are experimentally manipulated. Bacteria and guppies are just two examples of species that have been observed evolving in response to changing environmental conditions.
What are chondrichthyes?
Chondrichthyes are fishes that have a cartilaginous skeleton rather than a bony skeleton. They include such organisms as sharks, skates, and rays.
What is the most important structural difference between gram-negative bacteria and gram-positive bacteria?
The most important structural difference between gram-negative and gram-positive bacteria is that gram-negative bacteria are enclosed by two bilayers, a cytoplasmic membrane and an outer membrane. Between the two membranes lies a thin peptidoglycan layer that is linked to the outer membrane. In contrast, gram-positive bacteria have a thick layer of peptidoglycan. The cell wall of gram-positive bacteria is between 2 to 8 times as thick as the cell wall of gram-negative bacteria. The thinness of the cell wall of gram-negative bacteria affects the bacteria's ability to retain the crystal violet iodine complex of the gram stain.
What is cell fluorescence?
Fluorescence is luminescence caused by a pigment molecule. Pigments are molecules that absorb some colors of light while reflecting others. For example, green pigments, like those in leaves, absorb red and blue wavelengths of light but reflect green wavelengths. After being energized by photons, units of light energy, the electrons of some pigment molecules actually give off light as they fall back to their normal state. Chlorophyll molecules that play a role in photosynthesis have this ability, as do molecules found in several organisms, such as jellyfish. The body of the insect Photinus pyrillus, better known as a lightning bug, has the enzyme luciferase, which generates the chemical reaction that leads to a drop in the energy state of electrons, a reaction similar to that which occurs in a chlorophyll molecule, allowing the insect to glow.
What happens to proteins as we age?
A variety of molecular changes typify aging. For example, there is a slowdown in production of the proteins collectively known as collagen, that maintain the skin integrity. As the amount of collagen in skin decreases, the skin loses elasticity and begins to sag, bag, and wrinkle. The loss of elasticity can be demonstrated by gently pinching a fold of skin on the back of the hand of differently aged people. The skin of young children will immediately return to form when released, while that of an elderly person will take longer.
Who eventually showed that plant cells were totipotent?
In 1958, Frederick Campion Stewart, 1904 to 1993, a botanist at Cornell University, successfully regenerated an entire carrot plant from a tiny piece of phloem. Small pieces of tissue from carrots were grown in a nutrient broth. Cells that broke free from the fragments dedifferentiated, meaning that they reverted to unspecialized cells. However, as these unspecialized cells grew, they divided and redifferentiated back into specialized cell types. Eventually, cell division and redifferentiation produced entire new plants. Each unspecialized cell from the nutrient broth expressed its genetic potential to make all the other cell types in a plant.
Who developed the gram stain and why is it important?
Staining is a process in which a scientist colors a microorganism with a dye that emphasizes certain structure. The gram stain was developed in 1884 by Hans Christian Gram, 1853 to 1938. A Danish physician who worked at a hospital in Berlin, Germany. The gram stain is one of the first steps in the identification of bacteria, and is used to categorize bacteria into two major groups, gram-positive and gram-negative. When the gram stain is combined with other information on cellular morphology and biochemical characteristics, conclusive identification of an unknown type of bacteria can usually be made. The gram stain is the most widely used stain in microbiology.
What is a race?
The term race was originally used to describe subspecies. However, as genetic analysis of humans has shown, there is greater genetic variation within geographic subpopulations, races, than among the human population as a whole. In other words, there is so much genetic overlap between groups formerly designated as races that the term is meaningless and biologically indefensible.
What is the one gene one enzyme hypothesis?
In the 1930s, George Beadle, 1903 to 1989, and Boris F. Russey, 1901 to 1979, theorized that the variety of fruit fly mutations might be due to mutations of individual genes that code for each of the enzymes involved in a given pathway. Subsequently, Beadle and Edward Tatum, 1909 to 1975, performed a series of experiments with the orange bread mold Neurospora that elucidated the enzymatic pathway required by the fungus to produce a specific nutritional requirement, arginine. The researchers were able to create a series of mutants, each lacking in a different enzyme in the pathway. In this way they were able to piece together the sequence of events required for the production of arginine and thereby show where each mutant fit. The work of Beadle and Tatum provided important support for the one gene one enzyme hypothesis, which holds that the function of a gene is to produce a specific enzyme. Their work garnered a Nobel Prize in Physiology or Medicine in 1958.
What is a mermaid's purse?
Mermaids' purses are the protective cases in which the eggs of dogfish, skates, and rays are released into the environment. The rectangular purse is leathery and has long tendrils streaming from each corner. The tendrils anchor the case to seaweed or rocks, where the case is protected during the 6 to 9 months it takes for the embryos to hatch. Empty cases often wash up on beaches.
What is needed for DNA replication?
DNA replication is a complex process requiring more than a dozen enzymes, nucleotides, and energy. In eukaryotic cells there are multiple sites called origins of replication. At these sites, enzymes unwind a helix by breaking the double bonds between the nitrogen bases. Once the molecule is opened, separate strands are kept from rejoining by DNA stabilizing proteins. DNA polymerase molecules read the sequences in the strands being copied and catalyze the addition of complementary bases to form new strands.
Who extensively studied fly behavior?
Vincent Dithier, 1914 to 1993, spent most of his life researching chemical responses in insects. He wrote numerous articles and books, both for the general public and specifically for children. To Know a Fly, perhaps his most famous book, is considered one of the classic books in entomology. The book details physiological research on flies, particularly with regard to chemoreception, with a touch of humor. Aside from his career as a distinguished research scientist, he was able to popularize what scientists do and what draws people to science.
Who was Mendel?
Gregor Mendel, 1822 to 1884, is the founding father of experimental genetics. His work with the garden pea, Pisum sativum, was not consistent with the 19th century ideas of inheritance. Mendel was the first to demonstrate transmission of distinct physical characteristics from generation to generation.
What is the difference between primary growth and secondary growth?
Primary growth occurs in the tips of stems and roots in plants, thus increasing the length of the stems and roots. Secondary growth allows a plant to increase its diameter. The results of secondary growth form the division of a cylinder of cells around the plant's periphery.
How can speciation occur?
If a population becomes reproductively isolated, then individuals within the population will no longer exchange genetic material with the rest of the species. At that point, environmental factors, i.e., natural selection, will work on the genetic variation within that population until it has become a new species.
What different types of aggressive behavior do animals display?
Animals can show aggression through sound, e.g., growls, barks, trumpeting, sight, e.g., changing coloration, inflating body structures, and even scent. They can change the way they move, where they perch, or how much tooth enamel they display. For example, yawning among male mandrills is often not an expression of boredom but rather an opportunity to display their well-honed canine teeth.
What are the differences between prokaryotic and eukaryotic cells?
Eukaryotic cells are much more complex than prokaryotic cells. Having compartmentalized interiors and membrane-bound organelles within their cytoplasm. The major feature of a eukaryotic cell is its membrane-bound nucleus, which compartmentalizes the activities of the genetic information from other types of cellular metabolism. Generalized prokaryotic cell.
How much newspaper must be recycled to save one tree?
(135 to 40 feet, 10.6 to 12 m), tree produces a stack of newspapers 4 feet. 1.2 m, thick. This much newspaper must be recycled to save a tree.
What is special about the spores of leptosporangiate ferns?
Leptosporangiate ferns are the most common ferns in North America. The sporangia of leptosporangiate ferns arise from a single surface cell, are relatively small, and have a delicate stalk and a thin sporangial wall. The small number of spores per leptosporangium is a multiple of 4, varying between 16 and 512, most often 16 or 32, in homosporous species. Each plant is able to produce millions of spores because of the large number of sporangia per sorus and the enormous number of sori per leaf. One mature plant of the species Thlypteris dentate can produce more than 50 million spores each season.
What is ethology?
Ethology emerged in the mid-1930s and was first recognized as a subdiscipline of biology in Europe. Ethology differed from traditional biological studies of animals in that scientific principles were applied to the study of animal behavior, with practitioners using both field observations and laboratory experiments. The field was developed and first recognized as a science in Europe, where experimental conditions were kept as natural as possible.
What contributions did John and William Bartram make to botany?
John Bartram, 1699 to 1777, was the first American-born botanist. He and his son, William Bartram, 1739 to 1823, traveled throughout the American colonies observing the flora and fauna of the colonies. Although John Bartram never published his observations, he was considered the authority on American plants. In 1791 his son William 160 published his notes on American plants and animals as Bartram's Travels.
How is an ant colony organized?
Ants are the dominant social insects and, numerically, the most abundant. At any one time there are at least 1,015, 1 quadrillion, ants alive on the planet. In general, ant colonies contain a variety of castes, groups of individuals with a common job, like workers or soldiers, and a queen who is in charge of egg laying. For example, the largest and most aggressive workers make up the soldier caste, whose job it is to protect the colony against dangerous invaders. Because of the way that sex is determined among ants, males are haploid, contain one set of chromosomes, while females are diploid, 2 sets of chromosomes. This causes a close interrelatedness among members of a colony that is theorized as one of the reasons for the evolution of social colonies.
Why will my dog play fetch but not my cat?
Domestication occurred differently for cats and dogs. while dogs came to work closely with humans. Relying on them for food and shelter, cats maintained a more distant relationship. A dog plays fetch because it is an extension of the cooperative working hunting relationship she has with humans, while the cat has no such relationship.
Why is the yeast Saccharomyces cerevisiae important in genetic research?
Biologists have studied Saccharomyces cerevisiae, a yeast used by bakers and brewers, for many decades because it offers valuable clues to aid in the understanding of how more advanced organisms work. For example, humans and yeast share a number of similarities in their genetic makeup. The DNA present in certain regions of yeast contains stretches of DNA subunits that are nearly identical to those in human DNA. These similarities indicate that humans and yeast both have similar genes that play a critical role in cell function. In 1996, an international consortium of scientists from the United States, Canada, Europe, and Japan completed the genome sequence, all 12,057,500 subunits contained in the nuclear DNA of S. cerevisiae. It is the first eukaryotic organism to be completely sequenced. With their rapid generation time, yeasts continue to be the organism of choice. To provide significant insights into the functioning of eukaryotic systems,
How can genes become rearranged?
any rearrangement within a gene is a mutation, and such rearrangement is usually due to a random genetic accident. The mutation can be harmless and simply add to the variation within an organism's genome, or it can be harmful with dire consequences.
How many enzymes are there?
Approximately 5,000 enzymes have been named, but there may be a total of 20,000 or more. A metabolic pathway may require a whole complex of enzymes to complete hundreds of reactions.
Who was Rachel Carson?
Rachel Carson, 1907 to 1964, was one of the first to describe to the general public the consequences of chemical contamination in the environment. In her book Silent Spring, published in 1962, Carson exposed the dangers of hydrocarbons, particularly DDT, to the reproduction of species that prey upon the insects for whom the pesticide was intended.
What is a community?
Suppose you wanted to study not just the sparrows living in your backyard but Also the insects they feed to their young and the plants that those insects eat. The term used by ecologists to describe a group of populations of different species living in the same place at the same time is community. This is a concise way of describing the organisms likely to be affected by any change to the local environment.
Why are proteins among the most complex molecules?
Among the major bioorganic molecules, only proteins require up to 4 levels of structure in order to be functional. In addition, relatively slight environmental changes cause a shift in structural levels that may be sufficient to radically change the function of the protein. The structural levels of protein and the functions they provide are: primary, a polypeptide chain of up to 500 amino acids covalently bonded. Secondary, the formation of hydrogen bonds between nearby amino acids causes the polypeptide chain to twist and slash or pleat. Tertiary, distant amino acids form bonds and associations. In reaction to changes that occur in the secondary level. Quaternary, two separate polypeptide chains intermingle to form a molecule that has a larger, more complex structure than that found in the other protein levels.
What is a bioinvader?
A bioinvader is an exotic organism usually introduced into an ecosystem accidentally. These bioinvaders are non-native plants and often overwhelm the native species. Examples of bioinvaders include the kudzu vine. Kudzu was first introduced in the 1930s by the United States Soil Conservation Service for a good purpose to control erosion. Kudzu now grows uncontrolled in the southeastern United States, pulling down power lines and killing trees. Other bioinvader species include zebra mussels, Great Lakes, purple loosestrife, northern United States and Canada, and the Asian longhorned beetle, first reported in New York but now spreading into the Midwest.
How does smoking affect the DNA of lung cells?
Smoking appears to alter gene expression in lung cells. The proteins produced by the bronchial cells of smokers show increased synthesis of genes, which can eventually be carcinogenic. It appears that the expression of these important genes that control cancer development, cancer suppression, or airway inflammation varies according to the number of years spent smoking. The good news is that 2 years after a person stops smoking, his/her gene expression levels begin to resemble those of people who have never smoked.
How are birds related to dinosaurs?
Birds are essentially modified dinosaurs with feathers. Robert T. Bakker, 1945, and John H. Ostrom, 1928. Did extensive research on the relationship between birds and dinosaurs in the 1970s and concluded that the bony structure of small dinosaurs was very similar to Archaeopteryx. The first animal classified as a bird, but that dinosaur fossils showed no evidence of feathers. They proposed that birds and dinosaurs evolved from the same source.
What is a base pair?
The phrase base pair is a way of describing the length of DNA. Since most DNA is double-stranded, every nitrogen base containing nucleotide on one strand of the molecule is paired with a complementary base on the other strand. Thus, 10 base pairs refers to a segment of DNA that is 10 nucleotides in length and 2 strands in width.
What is meant by pH and why is it so important to living things?
The term pH is taken from the French phrase T puissance d hydrogen, meaning the power of hydrogen. The composition of water can be used to understand the concept of pH. Water is composed of 2 hydrogen atoms bonded covalently to an oxygen atom. In a solution of water, some water molecules will dissociate into the component ions, the H and ion and the O ion. In chemistry, the term equilibrium reaction is used to describe a situation in which there are an equal number of products and reactants. Water is an example of an equilibrium reaction. Water contains H2O, H+, and O at all times. It is the balance of the 2 ions that determines pH. When there are more H and ions than O ions, the solution is referred to as an acid. And when there are more O ions than H and ions, the solution is referred to as a base. The concentration of hydrogen ions in water influences the chemical reactions of other molecules.
What is Botox?
Botox, the trade name for botulinum toxin type A, is a protein produced by the Bacterium Clostridium botulinum. Although it is the same toxin that causes food poisoning. Purified botulinum toxin that is sterile and has been converted to a form that can be injected and used in a medical setting. Botox was first approved by the Food and Drug Administration, FDA, in December 1989 to treat 2 eye muscle disorders: uncontrollable blinking, blepharospasm, and misaligned eyes, strabismus. In 2000 the toxin was approved to treat cervical dystonia, a neurological movement disorder that causes severe neck and shoulder contractions. In small doses it is able to block nerve cells from releasing a chemical called acetylcholine, which signals muscle contractions. by selectively interfering with the muscle's ability to contract. Existing frown lines are smoothed out, improving the appearance of the surrounding skin.
What are the ideal conditions for fungi to attack wood?
Fungi tend to attack woods when temperatures range between 50 degrees and 90 degrees Fahrenheit, 10 degrees and 32 degrees Celsius. Wood needs to be moist for fungi to grow. The most serious decay occurs when the moisture content of the wood is approximately 30%. Wood with a moisture content of less than 20% will usually not decay, and any infection will not progress. Wood that is too wet will not decay because the excess moisture does not allow fungi sufficient access to air, thus impeding their growth.
Why is evolution a theory?
A scientific theory is an explanation of observed phenomena that is supported by the available scientific data. The term theory is used as an indication that the explanation will be modified as new data becomes available. For example, the Darwin-Wallace theory was proposed prior to the discovery of the molecular nature of genetics but has since been expanded to encompass that information as well.
What is junk DNA?
The non-coding portions of the genome were formerly referred to as junk DNA. Some of this material turns out to play regulatory roles in DNA replication and transcription, and the rest may be an evolutionary holdover.
What is eutrophication?
Eutrophication is a process in which the supply of plant nutrients in a lake or pond is increased. In time, the result of natural eutrophication may be dry land where water once flowed, caused by plant overgrowth. Natural fertilizers, washed from the soil, result in an accelerated growth of plants, producing overcrowding. As the plants die off, the dead and decaying vegetation depletes the lake oxygen supply, causing fish to die. The accumulated dead plant and animal material eventually changes a deep lake to a shallow one, then to a swamp, and finally it becomes dry land. While the process of eutrophication is a natural one, it has been accelerated enormously by human activities. Fertilizers from farms, sewage, industrial wastes, and some detergents all contribute to the problem.
What are heterosporous plants?
Heterosporous plants produce two types of spores, microspores and megaspores, which develop into the male gametophyte and female gametophyte, respectively. In 1580 the physician Prospero Alpini, 1553 to 1616, identified that plants exist in male and female forms.
What is the hydrologic cycle?
The hydrologic cycle takes place in the hydrosphere, which is the region containing all the water in the atmosphere and the Earth's surface. It involves 5 phases: condensation, infiltration, runoff, evaporation, and precipitation.
What is a better source of energy, fat or sugar?
Fats contain 9 kcal/g, while carbohydrates average about 4 kcal/g. Fats are useful for storing energy, but sugars are easily broken down during metabolism. Nutritionists and diet gurus are debating which source of energy is better. The absolute answer lies in individual situations. For example, babies require fat in their diets for both energy 24 and to build a healthy nervous system. Middle-aged adults quite often take in too much fat for their metabolic requirements. As one ages, average energy needs decrease by 5% per decade.
What are the different types of natural selection?
There are several ways in which natural selection can cause a population to change. Natural selection can cause a trait to change in one direction only. For example, when individuals within a population grow taller with each generation, this is known as directional selection. Diversifying selection can cause the loss of individuals in the mid-range of a trait. For example, if there is a range in color of a certain prey species from very dark to very light, those individuals in the mid-color range may not be able to hide from predators. The mid-color prey will then be selectively removed from the population, leaving the population with only 2 forms, the very dark and the very light. In stabilizing selection, those at either end of the range are removed more often, creating selection pressure for the mid-range. Selection can also work on traits important to sexual reproduction. This is known as sexual selection.
What is a hybrid?
A hybrid is produced as the offspring of 2 true breeding organisms of different strains, AAXAA. If the hybrids, AA, are then mated, the result is a hybrid cross.
What is the advantage of thin layer chromatography?
Thin layer chromatography is a much quicker technique than paper chromatography. To separate a mixture using paper chromatography may take 24 hours. But less than 1 hour using thin layer chromatography.
Which animals can run faster than a human?
The cheetah, the fastest mammal, can accelerate from 0 to 45 miles per hour, 64 kilometers per hour. In 2 seconds, it has been timed at speeds of 70 miles per hour, 112 kilometers per hour, over short distances. In most chases, cheetahs average around 40 miles per hour, 63 kilometers per hour. Humans can run very short distances at almost 28 miles per hour, 45 kilometers per hour, maximum. Most of the speeds given in the table below are for distances of 0.25 mi, 0.4 kilometers.
Why do puffer fishes puff?
Pufferfishes are any of a number of species found in warm seas that use a special adaptation of the gullet to inflate their bodies to nearly twice the normal size. Pufferfishes, blowfishes, and their like do this in response to a perceived threat. The increased size and unpalatable-looking spines make the potential prey look quite unappetizing to predators.
Do genes affect your mood?
Mood disorders are conditions that go beyond an occasional bad day into the realm of severe emotional disturbance. Mood disorders include depression, the most common mood disorder, bipolar disorder, and schizophrenia. By examining family medical histories and adoption studies, researchers have concluded that there is a link between bipolar illness and genetics. However, the presence of certain gene sequences is not a mandate, as only about 60% of monozygotic identical twins with the bipolar sequence actually become ill. It is obvious that environmental and societal factors also play roles in determining one's mood.
What are Joshua trees?
Yucca brevifolia, a large shrub found in the southwestern region of the United States. Received its common name from Mormon pioneers. They named the tree after the prophet Joshua because its greatly extended branches resemble how Joshua used his outstretched arms to point his spear toward the city of Ai.
Which mammals lay eggs and suckle their young?
The duck-billed platypus, Ornithorhynchus anatinus, the short-nosed echidna or spiny anteater, Tachyglossus aculeatus, and the long-nosed echidna, Zaglossus bruigini, indigenous to Australia, Tasmania, and New Guinea, respectively, are the only 3 species of mammals that lay eggs. A non-mammalian feature, but suckle their young, a mammalian feature. These mammals, order Monotremata, resemble reptiles in that they lay rubbery, shell-covered eggs that are incubated and hatched outside the mother's body. In addition, they resemble reptiles in their digestive, reproductive, and excretory systems, and in a number of anatomical details, eye structure, presence of certain skull bones, pectoral shoulder girdle and rib and vertebral structures. They are, however, classed as mammals because they have fur and a 4-chambered heart. Nurse their young from gland milk, are warm-blooded, and have some mammalian skeletal features.
Are there more marine or freshwater sponges?
There are approximately 5,000 species of marine, saltwater, sponges and 150 species of freshwater sponges.
Who was Konrad Lorenz?
Konrad Lorenz, 1903 to 1989, became famous for his work in the field of avian ethology, particularly in his studies of imprinting. By raising goslings from the time they were hatched, Lorenz was able to make the goslings follow him rather than their own mother. This work led to the theory that the goslings were genetically programmed to exhibit a certain behavior with regard to any large organism that was near them during a critical early period of their life. In his publications he applied comparative methods to the study of behavior and the psychology of perception.
What is a MVP?
The MVP, minimum viable population size, is the smallest number of individuals needed to perpetuate a population, subpopulation, or species. PVAs, population viability analyses, are especially helpful in predicting the MVP.
What do proteins do?
In a word, everything. Proteins allow life to exist as we know it. They are the enzymes that are required for all metabolic reactions. They are also important to structures like muscles, and act as both transporters and signal receptors.
What is the lifespan of a fruit fly?
The length of adult life can vary considerably. Under ideal conditions, an adult Drosophila melanogaster can live as long as 40 days. In crowded conditions, lifespan may drop to 12 days. Under normal laboratory conditions, however, adults generally die after only 6 or 7 days.
What does the term hydroponics mean?
This term refers to growing plants in some medium other than soil. The inorganic plant nutrients, such as potassium, sulfur, magnesium, and nitrogen, are continuously supplied to the plants in solution. Hydroponics is mostly used in areas where there is little soil or unsuitable soil, since it allows precise control of nutrient levels and oxygenation of the roots. It is often used to grow plants used for research purposes. Julius von Sachs, 1832–1897, a researcher in plant nutrition, pioneered modern hydroponics. Research plants have been grown in solution culture since the mid-1800s. William Garrick, a scientist at the University of California, defined the word hydroponics in 1937. In the 50 years that hydroponics has been used on a commercial basis, it has been adapted to many situations. NASA will be using hydroponics in the space station for crop production and to recycle carbon dioxide into oxygen. Although successful for research, hydroponics has many limitations and may prove frustrating for the amateur gardener.
What is high-pressure liquid chromatography?
High-pressure liquid chromatography, HPLC, was developed in the mid-1970s. It differs from other chromatographic techniques in that the liquid is pumped through a short packed column under pressure, instead of relying only on gravity.